Hemraj, are you writing the exam in 2022? If it is for 2022, probably I'll be starting another batch in the month of June, so you can continue in that. It's been already some 51 classes, 50 classes completed, so it's a little difficult for you to understand today. But if you have you're writing for 21, you can try to cover up the recorded classes and attend this. Yes, I heard that. That was obvious, of course. Not the date, but postponement was kind of. So, shall we continue? First dose of vaccine, congrats. So they are giving the first dose also, because they said first they'll complete the second dose for everybody. So, we were discussing human genetics yesterday, and we started with the pedigree analysis. Okay. And in the pedigree analysis, yes. So most of, I don't know with what plan they have sent the vaccines to the other countries. I don't know. With so much of vaccine production capacity, not even 1% people got vaccinated. So I don't know what is happening. Very big failure of the state. Yeah. Vitamin C, paracetamol, these are also not available these days. And the crowded the vaccination centers. They are the big super spreaders. So they should give some slots, and according to the slots, they have to do that. Don't know what happens, but they are saying that we reached the peak. So let us hope there will be a quick decline. Yeah. Okay. So now let us continue with the topic. We started the pedigree analysis, and in the pedigree analysis, second wave is not yet completed. Now they are speaking about the third wave. But I think this virus is actually quickly spreading in the summer seasons. So probably with the rainy season and winter, it will be a little less, then again it may shoot up. But in the gaps also, the people will not prepare the hospital facilities in infrastructure. They won't develop. In the gaps, they will enjoy and cry in the peaks. Yes, that is another big worry. Yeah. So let us proceed with the subject now. We have seen autosomal dominant characteristics. Now what you have to do is look at the autosomal dominant characteristics, and you tell me the characteristics for autosomal recessive. Means for every point, say I'll pick one point. For that, you have to tell me the point for. I hope the question is clear. You have to tell me the corresponding point for autosomal recessive. If it is recessive, what will be the point? So tell me the first point. We will have skipping of generations. Skipping of generations will be there. And skipping of generations means carriers, because skipping occurs in the form of carriers. Means because they are carriers, they will not have the disease. But as they can carry the disease to the next generation, again next generation will have the disease. So that is nothing but skipping. Okay. Then the third point, every diseased individual need not have a diseased parent, isn't it? Two carriers can give birth to a diseased child. Two carriers can give birth to a diseased child. So two carriers means both of them are normal. Carriers are normal people only. Okay. So like this, two carriers, they both are normal, but their child can be 1/4 chance that their child can be diseased. Okay. Then next point, the autosomal recessive characters are expressed only in homozygous condition. Means only homozygous condition means what? Capital A or small a? No. Capital A are not diseased. Capital A are not. One second, Anil. Okay, few people have given the wrong answer. Small a is the correct answer. Expressed only in small a. Capital A people will be normal. Why they are normal? Because if it is autosomal recessive, it means small a is the lethal gene. Capital A is innocent. It is not having any link with this. Did you understand? So here only homozygous condition means what? Only small a will express. Okay, capital A are not the diseased. Is it clear for those who said capital A and small a? Some people gave this answer. Is it clear for you? Okay, fine. Thank you. Next one, fifth point, for autosomal recessive. Remember, now we are discussing about autosomal recessive, right? Both sexes have equal chance of inheritance because that is autosomal. That is autosomal. Very good. Okay. Then now, next point, transmission among all sexes is possible. Mother can inherit the disease to the son. Father can inherit the disease to the son. Mother can inherit to the daughter. Father also can inherit to the daughter. Okay. Then this is one example pedigree. Okay. If you observe what points you have to observe. Look at here, shaded squares are there. Somebody wanted me to explain this. This is what pedigree? Autosomal dominant, isn't it? This is autosomal dominant. If you observe, there are no carriers. Do you have any half shaded squares and circles? No, that point is observed. Second, shaded squares are also there, shaded circles are also there. Roughly equal number. Equal number of males and females are inherited, isn't it? And there is no skipping of generations. 1, 2, 3, all the 3 generations, there is disease. We can observe all those points in this pedigree. Either you can remember and draw the same pedigree, or you can draw the pedigree of your own choice. Okay? If you understand the concept, you can easily draw the pedigree. Clear for all of you? Can we proceed? And the examples are Huntington's disease, acanthocytoma, polycystic kidney disease, brachydactyly, porphyria, polydactyly, etc. Now let us check what you said for autosomal recessive. The second one we are discussing is autosomal recessive. You told me the points for autosomal recessive. Let us check whether the points are correct or not. First, when two carriers mate, each child has 25% chance of becoming homozygous, that is unaffected. 25% chance of being homozygous, that is affected. Affected means diseased. 50% chance of heterozygous, isn't it? Let us put it in the form of a cross. When two carriers marry, Okay, then 25% chance of being homozygous unaffected, 25% chance of being homozygous affected, 50% chance of being carriers, being heterozygous or unaffected carriers. Is the first point clear? Then expressed only in homozygous condition, equally expressed in both the sexes. Skipping of generations is observed. Heterozygotes act as carriers for the disease. That is, they themselves do not suffer from the disease but carry the disease to next generation. Both males and females can be carriers, isn't it? Can we have both male and female carriers? Very good. Every affected individual need not have a diseased parent. To carriers, to unaffected people can give birth to a diseased child. Okay, so this is an autosomal recessive pedigree. Okay, if you observe here, they have shown skipping of 1, 2, 3. Skipping of generations is shown. Carriers are shown, and male and female both carriers are shown. How unaffected? Carriers means unaffected only, no? Affected means diseased. Here, affected means diseased. Okay, you will call them as carriers only if they are unaffected. Got it, Ramnaran? Okay, carrier word itself means unaffected because they are affected. You will call them as affected only. Why will you call them as carrier? Okay, affected means diseased. If they are diseased, call them as diseased. If they are completely normal, call them as normal. If they are normal, not affected by the disease, they are not suffering, but can carry the disease daily, then only we use the word carrier. Okay, so how affected carrier itself is a very fundamental question. Okay, more concentration please. Then how do we draw the pedigree diagram? Like this only. That is the reason why I have given the diagrams. No? You have to draw like this only. Are they difficult? Either copy as it is, okay, if you don't want to think, or you can draw your own pedigree. Okay. So they have shown skipping of generations, they have shown male, female carrier, affected people, equal number of males and females affected. So all the characters that we have seen, okay, is indicated in this diagram. Then affected individuals are indicated by solid black symbols and unaffected carriers are indicated by half black symbols. Next examples. These examples are more familiar. Sickle cell anemia, phenylketonuria. Do you remember sickle cell anemia we discussed later? And do you remember phenylketonuria? Can anybody tell me? Do you see phenylketonuria as an example for what? We discussed this as an example for some concept. Phenylketonuria plus alkaptonuria. These two we have discussed as an example for. Lethals. What type of lethals? Others please. Adish correct answer. Others please. Conditional lethals. Means they cannot digest phenylalanine and tyrosine. So if you don't eat, you are okay. But if you eat, okay, then it can lead to death. Okay, it becomes a lethal. But not everybody. If they have the disease, they should not eat. Fava beans is different story. This is not fava beans. This is phenylalanine and tyrosine amino acids. We should not eat the food. They should not. It means the people having those diseases. But of course, fava beans also we discussed as an example for conditional lethals only. But all conditional lethals are not recessive diseases. Okay, recessive diseases are different. Conditional lethals are different. Here, they became the examples for both. Right? Phenylketonuria, alkaptonuria, they are examples of autosomal recessive because small a will cause the disease. Okay. For example, phenylketonuria if you take, capital P, normal or diseased? All of you, phenylketonuria if you take. In the diagrams, if there are any mistakes, because these diagrams I have taken from some source, I did not draw them. So understand the concept. In the diagram, if there is any mistake, you can correct it, no problem. Okay, you draw technically what is correct. Explain to the concepts clearly. Okay, fine. So normal, fine. Capital P, small p. Capital P, small p, normal or carrier. Got it? No, they are not diseased. Ramnaran, somehow you are not listening. If there is any doubt, you can please ask me. Ramnaran, if you did not understand something, please ask me, I will repeat. This is a recessive disease. Recessive disease means capital A is hero, small a is villain. Do you understand? Capital A is innocent, small a is the villain. Okay, capital A people, capital A do not cause the disease. Ramnaran, is that clear? Because your previous answer, that is when previous question I said homozygotes are diseased. Then you said like I think capital is same mistake here also. Okay, so remember for dominant diseases, capital A is villain, small a is hero. Do you understand? Okay, that is the reason why they are called recessive. Capital A has nothing to do with the disease here. Okay, no connection at all. So capital B, capital B, normal, capital P, normal, small p, diseased. Small p, diseased. Same with sickle cell anemia also. Okay, capital S, normal, capital S, small is normal, but that is also an example for incomplete dominance. See, remember, a disease can be an example for 2-3 different concepts. Is it clear? Agree? For example, sickle cell anemia is an example for incomplete dominance and autosomal recessive diseases. Phenylketonuria, alkaptonuria are examples for conditional lethals plus autosomal recessive. Okay, then you have to understand both the concepts separately. Okay, then so sickle cell anemia, alkaptonuria, spinal muscular atrophy. Muscular atrophy means their muscles are very weak. They will not have the muscle strength, so they cannot stand properly. They keep shivering. They cannot hold anything. They cannot lift the weights. Okay, dystrophy means that only weakness, degeneration, weakness. Okay, then microcephaly, unusually small head. You know this albinism. Okay, lack of melanin pigment. Okay, lack of melanin pigment. Small a or albino. Then schizophrenia. But of course, for schizophrenia, because there are many other reasons also. Okay, there are multiple reasons for schizophrenia. Schizophrenia is split personality. There are multiple reasons for it. One reason is autosomal recessive inheritance. So autosomal dominant, autosomal recessive, clear for all of you? Can you proceed? Autosomal dominant, autosomal recessive. Okay. Next we have to see X-linked dominant, X-linked recessive. Okay, X-linked dominant, X-linked recessive. Okay. Now, first I'll explain X-linked dominant. Okay, and the only trick is don't miss words or sentences in between. Okay, then you will 100% you will understand. Okay, one only reason if you are not understanding is diversion of thought in between. Okay, then so now let us see X-linked dominant. But here we have to see males and females separately. Okay, and wherever you don't understand, stop me there itself. For X-linked characters, males are two types: X capital A Y, X small a y. Agreed? Okay, X capital A Y. It means what? So males have only one X chromosome and one Y chromosome. If the X is having capital A, then we call it as X capital A Y. Okay, or another possibilities. Okay, the X chromosome is having small a. Whatever the X chromosome is having, Y chromosome do not have the same allele. Then we write this as X small a y. Agreed? Y chromosome do not have that gene. So males are two types: X capital A Y, X small a y. Females are three types: X capital A, X capital A. Okay, X capital A, X small a, X small a, X small a. Got it? Y is same for both. Y has no relation at all. Y is not at all an actor in that movie. Okay, not hero, not villain, not even an actor in that movie. Okay, Y has nothing to do with X-linked characters. You tell me what is the relation here. Got it? So females can be three types. Agreed? One possibility is they both have X chromosomes. This we are writing is as X capital A, X capital A. Somebody, okay, Y, yes, okay, thank you. Fine. Okay, and another possibility is X capital A, X small a. We write like this: X capital A, X small a. Got it? Another possibility is both the X chromosomes are small a. Then we write it like X small a, X small a. Okay, and for your clarity, I have drawn them as images. Otherwise, actually, I'm supposed to just write like this, and I have again drawn them for your clarity, because you may not understand what is X capital A, Y, what is X small a, Y. So, got it? What we are writing like this? Repeat. Run around. You're thinking about some of your friends. Is there anybody disturbing you? Something going on in your mind. Okay. Okay, fine. I'll repeat once again. But little more concentration, please. Okay. Look at here. Males are XY. Agreed? Means males have only one X chromosome. Okay. Pratyush Ramnarayan, particularly for you both. So I expect you to respond, please. Okay. And like here, males have only one X chromosome. And we are discussing about what? X-linked characters. Means genes on X chromosome. We are speaking about the genes on X chromosome. Got it? So Y chromosome has nothing to do with this. Right? So, for example, if the males are XY, this is X chromosome, this is Y chromosome. If you remember, I said Y is a smaller chromosome. X is female's X chromosome, Y is male's X chromosome. So, for gene A, there are two alleles. So, this X chromosome in the male can either have capital A or can have small a. Right? So, this is, say, some Krishna. Okay, Krishna is having capital A gene on X chromosome. Rama is having small a gene on X chromosome. Got it? So, Krishna is X capital A, X superscript capital A. Means what? On X chromosome, he is having capital A. On Y chromosome, he is not having the gene A. Got it? This diagram I'm writing like this in writing. So, for Pratyush, it is here, Rama right here. Okay? There. So, and what about Rama? Okay, for Rama, he is having small a allele. Okay, so we write like X small a. Means on X chromosome, he is having small a. On Y chromosome, he is not having the gene A at all. Neither capital A nor small a. Okay? So, this is how we represent. Whereas if you take females, okay, and one small request, okay, or one small general statement for everybody, okay, for the entire batch, okay. So, basically, I want you to answer the questions. That is the reason why, like, I'm so many times telling wherever you get the doubt, there itself you can ask me, and I don't consider any doubt as a silly doubt. And to give you confidence, I also challenge you that you cannot teach me some engineering or mathematics, okay. So, it means what? We all have a particular background, we all have a particular mindset, okay. And say, for example, if I joke, like, maybe your thought is getting diverted, okay, is it something unusual? No, that happens for everybody, okay. Because in a scientific experiment, what they have proven is, let us suppose a person is having a lot of, like, he loves ice cream so much, okay, and eagerly waiting to eat ice cream so much. But if you give him ice cream, 2 minutes without thought diversion, he cannot focus only on the ice cream. Do you know this? This is a psychological experiment. An ice cream lover, after so much of starvation, if I give him an ice cream, for 2 minutes, he cannot think only about ice cream. So if I joke upon something that is your thought is getting diverted, that is not unusual. That happens to me also. Sometimes while teaching also the thought will get diverted. You know that? Not only while listening. Okay. And I also said that as a student, I had the least attendance. I could not focus on the class at all. Okay. And my focus time duration is very, very little. So I don't want anybody to get offended. That's only a joke. Second thing, you can just consider me as your friend. Okay. So you can also joke upon me. I know you do that. Okay. So if I joke upon you, you should not take it serious. This is only just to have some conversation with you. That's all. Okay. There. So for females, if you take, they have two X chromosomes. So on one X chromosome, if they have capital A, on the other, if they have capital A, then we write it as X capital A, X capital A. Okay. Then if the second one, if they have small a, then we write this as X capital A, X small a. Got it? Then on both of them, if she is having small a, then we write it as X small a, X small a. So the same thing. Okay. So meanwhile, I have erased the thing that I have written. We are discussing about X-linked dominant characters and males, females. Okay. Males are X capital A Y or X small a Y. These are two possibilities. For females, there are three possibilities: X capital A, X capital A, X capital A, X small a, X small a, X small a. Got it? That's what by mistake I'm doing it. Flow. Okay. Now tell me, this is X-linked dominant disease. Okay. It means capital A is the lethal here. Capital A is the disease causer. Got it? Right. So here now tell me, X capital A Y people, are they diseased, normal, or carrier? I have given three options: diseased, normal, carrier. Very good. Diseased. X small a y, same three options. Deceased, normal or carrier? Normal. Means, do you agree male cannot be, okay? But of course, this is dominant, so there won't be a carrier at all. This is dominant, so no carriers. Okay, I'll come to that point later. Now let us see. Okay, look at here. X capital for female, same three options. Normal or deceased or carrier? Very good, deceased. Very good. Second one, this one, for this one. Normal or deceased or carrier? Carrier option also is there. Normal, diseased, carrier. I have given three options. Got confused. I wanted to confuse you. No carriers, because this is dominant disease. Do you remember one fundamental point? That is the reason why I stressed carrier also. Immediately carrier to 3B. Okay? So, fundamental point is, dominant allele will express in heterozygous condition also. So, when you have one dominant allele that is sufficient, they are also diseased. Okay? Then here, normal or carrier or diseased? Carrier. Carrier option is also there. Normal only. You don't want to get confused. Yes, in recessive carrier is there, but we are dealing with the dominant disease, right? Okay, I will draw one red line also. Ramnarayana, is that clear? In recessive carrier is there. Okay, but this is dominant. We are now discussing about X-linked dominant. Okay? So these are normal. Fine. So now tell me, what is the chance of a male getting such disease? Percentage. What is the chance of males getting this disease? Very good. Very good. What is the chance of females getting this disease? 75. And don't forget this. So, who are more affected with X-linked dominant diseases? Females have a higher chance of getting X-linked dominant diseases. Okay? Now, give me a few answers. Do we have carriers here? Very good. Means, do we have skipping of generations here? Very good. Should every deceased child have at least one deceased parent? Every deceased child have at least one deceased parent? Very good. Okay. Are males and females equally affected? I'm giving clue. Are males and females equally affected? Very good. So who are more affected? Females. Done. X-linked recessive, all characteristics you told me. You only told me all characteristics for X-linked dominant, I mean sorry. X-linked dominant. So let us cross check whether your points are correct or not. Okay. Because gene is located on X chromosome, there is no transmission from father to son. Agreed? Father cannot carry the disease to the son. Why? Because son is XY. Okay, son is XY, son is male, right? So, he will get one chromosome from the mother, one chromosome from the father. So, mother cannot give Y chromosome. So, definitely X chromosome came from the mother. So, Y chromosome is coming from the father. So, son will get Y chromosome from the father. Whereas a daughter, okay, will get one X chromosome from mother, one X chromosome from father. Isn't it? Can a son get X chromosome from father? No, he will get Y chromosome from father. So, can I say this point as it is will come in X-linked recessive pedigree also? Same point will come in X-linked recessive pedigree also? Very good. As the gene is located on chromosome, there is no transmission from father to son. But there can be transmission from father to daughter. Agreed? There can be transmission from father to daughter. Then, all daughters of an affected male will be affected since the father has only one X chromosome to transmit. If there is an affected father, all his daughters will be affected. Clear? At any point if you have a doubt, ask me there itself. Next. Children of an affected woman have 50% chance of inheriting the X chromosome with the mutant allele, right? Children of affected women have 50% chance. This one let us check once. Affected woman means what? Affected woman means it can be X capital A or it can be X capital A, X small a, right? Okay. If it is X capital A, okay, then all her daughters will get the disease. Can we say like that? All her daughters will get the disease. Okay. But if it is like this, 50% of her daughters will get the disease because each daughter may get either this or this, right? 50% of her daughters will get the disease. Okay. So, like that, in this point, if there is a children of affected women have 50% chance of inheriting the X chromosome with mutant allele. But actually, this point is applicable only for heterozygote. Isn't it? Children of affected women have only 50% chance of inheriting the X chromosome with mutant allele. This point is correct, but this is applicable only for heterozygote. So, you can write this and you can write a point about homozygote also. It means you can add one point there. Clear? Children, therefore, son and daughter taken together. Okay. Yeah, I'll repeat. But which point you want me to repeat? Because we have to take the other partner also. Okay. Let us check here. See, in genetics, if you don't understand something, you just have to sit with that. Solve it like a puzzle. For example, you have Sudoku. Okay. The moment you see that, you will not get it. If you start doing it, you will get it. Right? So, these are all very simple. If you don't get it, okay, just, for example, even I did not understand that point. So, what we have to do? Draw the cross and check it. Okay. So, if the woman is, affected woman is the point there. If the affected woman is X capital A, what about her daughters? Daughters, 100%. Okay. What about her son? Son also 100%. Son also 100%. Right? Reason is, let us suppose if the son got only one X chromosome from the mother, X capital A Y, then he is also deceased. If you are writing in 2022, you can wait for the next batch. Okay? If you're writing in 2022, then you can wait for the next batch. If you're writing in 2021, then quickly complete the recorded classes. It is important to follow the order. Okay? So, better you start with the recorded classes in that case. Then I have plans of starting another batch in the month of June. Okay, coming to this. Is it clear? Daughter also 100% chance, son also 100% chance. Okay? Okay, if this point is confusing you, leave this point, leave that point. Okay, look at here. Write this in the form of a point on your own. Got it? You need not write these points as it is. They are not quotations by a thinker. So if that exact sentence is confusing, ignore that. Okay? You can even, you need not even put it in the form of a sentence. It will be more effective if you draw like this also. Okay, you can write like this only. It's not that you have to write it in the form of a sentence only. Got it? So what about her daughters? Her daughters, okay. 50% of her daughters, because a daughter can either receive this chromosome or that chromosome. If she receives this chromosome, whatever type of X chromosome comes from the father, she will be deceased only. 50% deceased. Whatever may be the other X chromosome from the father, because one dominant allele is sufficient. Same if you take sons. Son will take only one X chromosome. Okay. So here also 50%. For daughters, kind of it also depends on who is the father, what X chromosome is coming from the father. Okay. So if the statement is confusing, you can write like this itself. Can we proceed? Because if the daughter gets this one, and from the father if she gets X capital A, then she can be deceased. Do you understand? So for daughters, it also depends on what is the genotype of the father. Not 100%, 100% for homozygotes. There is a chance of 100% also. Where? Krishna Kumar, how? For homozygotes, 100%. For heterozygotes, 50%. There is no question at all. I am just explaining something. Okay, the question is affected woman. What is the chance of her children being affected? X small a from father, X small a from mother. He is not deceased. Yeah, not deceased. If daughter gets capital A from mother, capital A from father, deceased. If daughter gets X capital A from the mother, okay. If the daughter gets X capital, listen these two statements clearly. If the daughter gets X capital A from the mother, whatever X chromosome comes from the father, she is deceased. Got it? Anuja is saying it depends on father. For daughters, it also depends on father. If she receives X capital A from the mother, then whatever X chromosome she receives from the father, she is deceased. But if the daughter gets X small a from the mother, then from father if she gets X capital A, deceased. From father also if she gets X small a, then she is normal. Got it? Clear for all of you? Avanija, Ananya. I just repeat once again. If she gets X capital A from the mother, then whatever X chromosome she receives from the father, she is deceased. But if she receives X small a from the mother, then if father also gives X small a, then she is normal. If the father gives X capital A, then she is deceased. Got it? This statement which is given here is not fully complete. That is correct only in certain conditions. That is only for heterozygotes. So instead of this sentence as it is, you can write on your own, or you can just draw like this. You need not even put it in the form of a sentence. Got it? Clear? Avanisha, if she gets capital A and capital A, then also we will say 50%. No, why then 50%? 50% is, we are speaking about all daughters, whatever daughters she will have. If she gets capital A from the mother, capital A from the father, then she is deceased. Okay, Ram Narayan, what is that 100? Wherever in dominant capital A is present, person is deceased. Yeah. See, many times I told you this point. If it is a dominant disease, capital A is, that is the reason why to fix in your mind I used the word hero and villain also. If it is dominant disease, capital A causes the disease. If it is recessive disease, small a causes the disease. Got it? That should be 100% or 50% in case of capital A and capital A. Capital A, 100%, right? Ram Narayan, if it is capital A, 100%. Got it? See, two instructions. Look at the slide, look at me, and complete concentration. Okay? Because sometimes I'm writing it clearly there and you're asking it. Means you're not seeing it there. For daughters, it is not straightforward like sons because it depends on what chromosome they are getting from father. Okay? Because they get X chromosome from both mother and father. It's not like it's not simple. The point is it will not depend only on the mother. Got it? Can you proceed? Then, affected female if homozygous can carry the disease to all her progeny. Clear? Affected female if homozygous can carry the disease to all her progeny. Like this. Again, I have erased. Sorry. Okay? Affected female if homozygous means if the female is X capital A, then 100% progeny means what? Male, female, both. Okay? Are affected. Then, and if you observe the pedigree, in the pedigree is skipping of generations shown? No skipping of generations. Every affected individual should have an affected parent, at least one affected parent. No carriers. No carriers. Okay? And actually, in this diagram, one point is not clearly shown. Do you remember 75% chance females will get affected, 50% chance males will get affected? So you have to show the shaded circles more in number. When you draw it, remember this point. But it does not mean that this is wrong. This is correct only. Okay? Because probabilities, for example, if I toss a coin only 2 times, is there a chance that like definitely one time I should get heads, one time I should get tails? And if the probability of getting the son means 50%, so is there a rule that every family should have one son, one daughter? No. So here if the 75-50 is not indicated, it does not mean that this is wrong. Means the larger the pedigree, the probabilities will come more accurately. Agreed? Father is deceased, son is deceased. Father is deceased means X capital A Y. Then mother is normal, so she is X small a, X small a. Son will get Y chromosome. So this is wrong. Yes, this is wrong. Okay, what Priyadarshini and Himesh pointed out is correct. This is wrong. If the father is deceased, mother is normal, son cannot be deceased. So actually, if you just put it like this, then the 75-50 also works out. Just correct that. Then the pedigree will cover the last point also. Got it? This is not something I have drawn and have taken it from source. So you basically understand the concept and you can draw your own pedigrees. Don't depend only on this. And it's not possible also. How do you remember this and draw? Drawing on your own is much easier. This is only to provide some examples. It's for affected female. What is for affected female? Okay, can we proceed? Is X-linked dominant pedigree clear for all of you? Then examples. Retinitis pigmentosa. This is some problem in the retina, so they cannot see the colors properly. Okay. And chondrodysplasia punctata. Chondrodysplasia punctata. Chondrodysplasia punctata. See, disease names actually 2-3 names if you remember, that is sufficient. This is some disease related to bones and cartilage. One thing, for diseases you need not write the symptoms here. Second one, you need not write the entire list also. So 2 to 3 examples are sufficient. Mainly you will write characteristics and the pedigree. Moreover, they will not ask you, till now they did not ask you to write down X-linked dominant pedigree. Do you understand? They will ask you a question on pedigree analysis. Then you have to write about all the 5 types of pedigrees. How many points you can write? Got it? How to remember these many diseases. That is the reason why we are discussing previous questions. When we discuss the previous questions, you have to understand, okay, how much you have to remember. They have never given a question right about X-linked dominant pedigree. They have given a question only on human genetics or pedigree analysis, man family study. Such questions were given. Then you have to write about all of them. So, in fact, 1 or 2 points not having 100% clarity, or some 30-40% of these points you forgot, still you can write excellent answer. Did you understand? So next, hypophosphatemic rickets. This is also the bones will become soft because of less phosphate. Hypophosphatemic rickets. Chondrodysplasia punctata. Retinitis pigmentosa. 1 or 2 examples sufficient. So can we proceed to X-linked recessive now? We are discussing X-linked recessive pedigrees now. Okay? X-linked recessive. Look at here. Read out the points. The gene is on X chromosome. There is no father to son transition, but there is father to daughter and mother to daughter and son transition. Is this point clear? Same point as X-linked dominant. Then, if a man is affected with X-linked recessive condition, can you tell me the genotype of affected man for X-linked recessive characters? I'm so carefully asking the question. I want all of you to give me the right answer. Genotype for affected man for X-linked. Okay. First I will talk to you individually. Say, let us take Himabindu's answer. Raj. So I am talking to you. Do you remember we are discussing X-linked recessive characters? Or probably people also. People, Raj, Himabindu. I am talking to you 3 people. Did you follow that we started X-linked recessive characters? We are not speaking about X-linked dominant characters. Okay, fine. That is fine. People, okay. Then other 2. Raj and Himabindu. Did you realize? This is X-linked recessive. So, the genotype of affected man, okay. For X-linked recessive characters, the genotype of affected man is X small a y. X small a y. Man is affected with X-linked recessive. All his daughters will inherit 1 copy of the mutant allele from him. Correct? All his daughters will inherit 1 copy because daughter will get X chromosome from the father. Daughter cannot take Y chromosome because if she takes Y chromosome, she will become son. So daughter definitely takes X chromosome from the father. So his daughter will inherit 1 copy of the mutant allele from him. Clear? Is this point clear? Now coming to second point. Predominantly males are affected. Let us check that point. Okay. Draw like this. Draw it along with me. Males, females. Do you remember males have only 2 genotype possibilities? There are only 2 genotype possibilities. X capital A y, X small a y. Isn't it? Genotypes are same here. Isn't it? X-linked dominant, X-linked recessive. Genotypes are same. But the phenotypes are different. Females have 3 genotypes. X capital A, X capital A, X capital A, X small a, X small a, X small a. Right? Clear till now? Do you agree? Do you remember the same genotypes we have written for X-linked dominant also? Because genotypes are the different letter combinations possible. That will be the same. But that genotype gives which phenotype is the different point there. Got it? Say, if it is X-linked recessive, I want all of you to observe this word, please. I'm highlighting it. Means small a is our villain here. Got it? Then, what is the phenotype of this man? What is the phenotype of this man? Exactly a Y. Very good, normal. Because actually has nothing to do with that disease. No relation at all. Small a will cause the disease. So, diseased. Can we have carriers here? Can we have a male carrier? So, male is always, listen to the statement clearly. For X-linked recessive characters, male is either normal or diseased but cannot be a carrier. Okay? Agreed? For X-linked recessive characters, male is either normal or diseased but cannot be a carrier. Why? What is the reason why he cannot be a carrier? Why he cannot be a carrier? There is a word for it. There is a word for it. Yes, Priyadarshini correct. Priyadarshini correct. Hemizygous condition. But carriers are heterozygous, isn't it? These are carriers. A carrier is heterozygous, but we don't have heterozygotes in the males. Very good. Fine. For females, exactly. Very good. Normal. It's small a. Disease. Because small a is our villain. Small a causes disease. Clear? So, can we have carriers here? So, we have carriers, but carriers are only females. Because that is the reason why in pedigree this symbol is indicated. Do you remember? But we don't have such a symbol. Do you remember? And I hope you have written this information. See, if I am reading out such a slide, you don't have anything to write. But if I am explaining something in a plain slide, better write along with me. Okay? Or immediately after I explain once. Such points also may be there in the notes, but not 100% guarantee. Okay? It may be there in the form of a sentence, but I write it in the form of an equation or a flowchart or a diagram or something. So if I am explaining something by writing on a plain slide, make sure that you copy that. At least take a photograph in your mobile. So, can you proceed? Okay, then. Okay, next one. So, it means, what is the percentage of females affected? What is the percentage of females affected? 25%. Very good. What is the percentage of male? One second, some mistake here. Ram Narayan, if you have any fundamental question, yesterday's question also, you can ask me. Because what I can sense is, somewhere some crucial point you missed. Were you there? Did you attend my previous genetics classes before the break? That is the reason. So please complete them first. See, I could sense it. Okay, so that is the reason. So please quickly attend the recorded classes, at least genetics classes. Okay, then next. So it is 25%, not 50%. Okay, for females. Why? Because this is normal. This is because the ratio is 1:2:1. This 1:2:1 story is discussed in previous genetics classes. Okay, so that is the reason why you are getting confused. Okay, so females 25%, males chance 50%. So complete my sentences. For X-linked dominant characters, listen carefully and answer. No hurry. X-linked dominant characters. Okay, we are right now discussing about recessive, but I'm comparing the two and giving you a question. For X-linked dominant characters, males or females, who are more predominantly affected? For X-linked dominant characters. Very good, wonderful, very good. What are the percentages? 75 to 50. 75 for female, 50 for male. Male is always 50. Let it be X-linked dominant or X-linked recessive. Male is 50%, isn't it? Because there are only two genotypes. Okay, if it is dominant disease, then this is diseased. If it is a recessive disease, then this one is diseased. But 50% chance. Okay, then for X-linked recessive characters, males or females, who are more predominantly affected? Males, because males have 50% chance. Females have only 25% chance. Got it? Okay, so now next point I have to explain is crisscross pattern of inheritance. Okay, look at this crisscross pattern of inheritance. Means grandfather to grandson through carrier mother. Grandfather to grandson through carrier mother. Okay, and see one small instruction. I think it is better for me to give this instruction in every class. If there are any students who have joined late, okay, if you are writing in 2022, no hurry. Roughly in the month of June, I will start a new batch. Then you can freshly begin. You will feel 100% comfortable. Or second option you can consider is listen all recorded classes only, no problem, okay, because almost all the doubts, all the possible doubts are being discussed, okay, because I myself raised so many questions and I will answer them, okay. Then you will have a continuity, okay, because this is something like a serial, you will start from the 30th episode, and understanding will be difficult, okay. So we may on the whole get it, okay, what is happening in total story, we may on the whole get it, but not as clearly as the person who is watching from the first episode onwards. That is second alternative. Third alternative is, okay, it means, for example, right now genetics class is going on, at least make sure that earlier genetics classes are completed. Okay, first complete genetics classes and then for or social theories are going on. Then at least first make sure that you complete theories, then you will have a continuity. So from the schedule, if you can understand such things, that is another alternative. Okay, yes, fine. Now I have to explain another point. Crisscross pattern of inheritance observed. Crisscross pattern of inheritance is observed. Okay, means what? Inheritance of the disease from the grandfather to the grandson through carrier mother. This is called a crisscross pattern of inheritance. Okay. No, females always do not dominate others because if it is X-linked recessive, then males are more predominant. Okay. So grandfather is deceased, grandson is deceased, mother is carrier. The disease is going from grandfather to grandson through carrier mother. Means you cannot have a carrier father, agreed? You cannot have a carrier father, agreed? So that is called crisscross pattern of inheritance. Means skipping through carrier mother only. That is called crisscross pattern of inheritance. Skipping can happen, but it cannot happen through, skipping means carrier. Do you remember that? If a generation is skipped, the skipping happens in the form of carrier. Okay, but not male carrier. What recessive only? Of course recessive only. I remind you we are discussing about recessive now. We completed dominant. Right now we are discussing about recessive. Okay, and one more thing, like a small general discussion for 2 minutes. As a student, what I faced is, I rarely used to listen to classes. If I listen, I used to get it out what they are discussing. Actually, it means which topic is going on. Okay, why they are saying this particular point. I remember that. So that is the reason why in between I keep reminding you. I remind you that we are discussing this point. Did you notice? That is for that. Because that is what I faced. I don't know about you. Okay, but in between I remind you that this is the point we are discussing. That is from that background. So it means a bad student can be a reasonable teacher. That is what I understood. So all problems I faced as a student, I remember and I try to not give you those problems. Mother is from another family. Yeah, basically, here the way you have to look at it is like not family. Like it means it's not like patriarchy, matriarchy, not like that. Is it coming through inheritance or is it coming through marriage? That is the point. Okay, is it coming through inheritance or is it coming through marriage? Okay, here grandfather in this context means her father. Do you understand? Her father. So here the point that is different from social and biology is because if it is matriarchy, father is coming from other family. If it is a patriarchy, mother is coming from other family. So that is not our issue of concern here. Our issue is it coming through inheritance or marriage? Here grandfather is not her husband's father. Okay, like her father. From her father she got it. She is inheriting it to her son. Means biological inheritance, not through marriage. Got it? Then. So that particular concept is known as crisscross pattern of inheritance, particularly having a carrier mother. Okay, deceased grandfather, deceased grandson, but mother is a carrier. That is called crisscross pattern of inheritance. Got it? So you can indicate crisscross pattern of inheritance like this. Say her father is deceased, means X small a, Y grandfather. Okay, then X capital A, X small a, mother. Okay, then X small a, Y grandson. This is called crisscross pattern of inheritance. And I know that you may get one doubt here. Listen to the statement carefully. Crisscross pattern is not a compulsion. Means it's not that you have to surely observe the crisscross pattern. Okay, because what if the grandfather is not affected? What if a granddaughter is born and the grandson can also receive X capital A from the mother? Is it clear? These are all the possibilities. So is there a rule that crisscross pattern will happen? No, it can happen. That's all. That is one possibility. Okay. So in the crisscross pattern, the point that you have to highlight is only carrier is mother, not having a male carrier. That is crisscross pattern. Got it? It is a characteristic feature of X-linked recessive diseases. Next, X-linked recessive disorders examples. Duchenne muscular dystrophy. This is also another type of muscular dystrophy. Hemophilia A. Do you remember clotting factor number 8 question? Hemophilia A. The severe combined immune deficiency disorder. Severe combined immune deficiency disorder. Corrected? Means the word deficiency did not come here. So severe combined immune deficiency disorder. Some form of congenital deafness. Congenital means by birth. Red-green color blindness means they cannot see red and green colors. G6PD deficiency. G6PD means glucose 6-phosphate dehydrogenase. G6PD means glucose 6-phosphate dehydrogenase. Yes, there are other types of color blindness. Complete color blindness we have. Okay. Favism need not be genetic. Oh yeah, this is related to favism, right? This is related to favism. Those who have this should not consume fava beans. Yeah. Clear for all of you? Okay. The last type of pedigree that we have to see is Y-linked inheritance. Y-linked, okay. X-linked recessive. This is X-linked recessive pedigree. This is X-linked recessive pedigree. If you notice, right here, and if you want, you can draw it like this also. Okay. And if you feel this particular pedigree is not completely informative, if it is not involving all the points, you can draw your own pedigree. Not only in the male. What is only in the male? Yeah, Y-linked. Okay, yeah. Y-linked means only male. Okay. So we completed autosomal dominant, autosomal recessive. X-linked dominant, X-linked recessive. Okay. Autosomal dominant and recessive are equal in males and females. X-linked dominant is more in females. X-linked recessive is more in males. Clear till now? Whereas Y-linked is only in males. All the 5 summary I have given. I repeat, autosomal dominant and recessive, equal chance of inheritance in males and females. X-linked dominant, females have higher chance of getting the disease. X-linked recessive, males have higher chance of getting the disease. Y-linked, females have no chance of getting the disease, only males. Clear? That is the reason why they are called as all-andric characters, all males. Polandric characters. All males. All deceased people are males. Therefore, it is called Polandric character. Got it? They are found only in males, not found in females, as they do not have Y chromosome. Example, hypertrichosis inne. Did you notice some males, especially in their 40s, 50s, they have hair on the ear? Here they will have hair. That is called hypertrichosis inne. Did you observe any female in their 50s or 60s having hair on the ear? Your father, many of your fathers probably, but any of your mothers? No. Because it is a bilingual character. That is a bilingual character. In males, it is quite common. Almost many males, old males, they have it. In their 40s, 50s, they have it. So hypertrichosis inne means external ear. It's not a disease, it's a condition. Disease means what? Discomfort in that way. Disease, I told you the meaning of that. Disease means disease. If that is uncomfortable, it is a disease. Okay? But it's not in serious condition, no problem with that, except having hair. Okay. So that's all that we completed pedigree analysis. Pedigree analysis completed. And it means human genetics completed. Okay. Then now I will remind you one more point. Okay. Do you remember there was a question, applications of Mendelian genetics? Applications of Mendelian genetics. Can I say that we could understand this autosomal dominant, autosomal recessive? All these are the characters given in Mendel's laws only, isn't it? Law of dominance, law of recessive, and law of dominance. Dominant and recessive values are defined by Mendel in his second law, law of dominance. Without that, all these concepts, that 1:2:1 ratio is from Mendel's laws. So two important applications of Mendel's laws are Hardy-Weinberg law, pedigree analysis. Means human genetics. Agreed? So can you now write a better answer for applications of Mendel's laws? There I just told you that it will be used in pedigree analysis, but this is how it is used. Can I proceed? So next topic is twin studies. Twin studies. Okay, let us see the syllabus of 9.1 months. Human genetics, methods and applications, title. Methods for study of genetic principles in man, family study, pedigree analysis. This we have completed. Till here we have completed. And in fact, maybe you are getting the notes in the form of slides, but they are not actually the slides. That is only the format. This is complete notes, syllabus, previous questions, everything. Okay. So after that, now we are discussing twin study, foster child, code-win method. This is what we are discussing right now. Okay, or let us do one thing, because for twin study today time will not be sufficient. So look at the question. Briefly describe various methods used in genetic study of man. See, this is how the questions will be. So how much you have to understand about pedigree analysis? What you have understood is too much to write this answer, isn't it? So 1 or 2 points are not very clear, nothing to panic. Different methods used in genetic study of man. What are the different methods used in genetic study of man? Hardy-Weinberg law, pedigree analysis. Isn't it? And you have to write the introduction that I have given in the beginning of yesterday's class. Got it? Man and others which we will study. Why do we study others? Our subject is anthropology, study only about man. What others we should study and understand? Adish, include no syllabus, other types of methods. Other types of methods, Hardy-Weinberg law or pedigree analysis, both, that's all. For man genetic study, these are the only 2 methods. Human genetics is different. See, this is one very important point. Human genetics is different. Genetic study of man is different. Okay, so when it comes to, like, there is another type of question that is related to biotechnology, applied molecular techniques. Okay, so genome studies and all. In that way, I will give you another answer later. That comes after we complete unit 12. As of now, this is what we have to see. Okay. Did we also complete this one? Human genome project? Did we complete that? So for this answer, okay, do one thing, write down this checklist. For this answer, you have to write human genome project. Okay, in the order. Okay, I'll give you this question we are discussing and I'm giving you the structure. Take down, write down. Okay, first you write the introduction. Introduction that I have given, that is controlled mating experiments are not possible. Do you remember? That you have to write. Then as a solution, we have 2 alternatives. One is Hardy-Weinberg law, that you have to write. Second one, pedigree analysis, that you have to write. Then third one, under advanced techniques, you can take another subheading as advanced techniques. You can also write about human genome project. This is how to study the human genetics. Then in unit 12, there is another topic. Do you remember recombinant DNA technology? In unit 12, they have given recombinant DNA technology. That is different. Okay, for that I will give you answer after unit 12. This is how to study the genes, not how to do genetic experiments in man. Okay, that is genetic engineering in man, recombinant DNA, how to, for example, design their babies, all that. That is how to do genetic experiments on man. This is how to study the genetics of man. Okay. So that is a different topic, different question, but that is also there in our syllabus. We will discuss that. We have it in our syllabus. If you are in SNT batch, okay, then in SNT, in fact, we do it more clearly. Okay, because here there is only one topic there, that is what we study. This is here, it is just one word given. So biotechnology aspect, okay, if you are also attending SNT classes, that is better. then you know that there is human genetic experiments, you know, if you have already attended the SNT classes. But Himesh, I didn't see you in that class. Adish also not regularly, right? Adish was there in my first batch, right? First SNT batch he was there. Recording. New batch is starting on 24th. New SNT batch will start on 24th. And by the way, if there are any of your friends, you can inform my referral code is Himabindu. My referral code is Himabindu. And I'm teaching anthropology optional that you know very well. In addition to this, I also teach SNT. SNT is not only biotechnology, complete science and technology. Okay, that is space technology, defense, nuclear technology, ICT. New batch is starting on 24th May. Okay, time is 9 a.m. Time is 9 a.m. How many classes? I think 22 classes. 22 classes. 22 classes. That is entire science and technology. Very, very important. People usually don't give much importance to science and technology, but average 12 questions are coming from class duration 2 hours. 2 hours, 22 classes. Okay, that is detailed. But in fact, you know my problem, right? I cannot move forward if you don't understand. So in fact, there is no much difference between crash and prolonged. Okay, both are equal. Current affairs, SNT means current affairs only. If you remove current affairs, there is nothing in SNT. Because in SNT, they are asking only from the current events. That includes biotechnology. Around 8 classes in that are biotechnology. Remaining all are other technologies. So biotechnology will be useful for anthropology also. Of course, in anthropology also we will discuss, but not so much in depth. Anthropology new batch timings are not yet fixed. Okay. And to extend this one, because they have given the word crash course, so to make it a little lengthier, I'm planning to include some MCQs in that. No, whatever I teach, I start from the scratch. So you need not come prepared to my classes. That is one thing I always follow. Whatever might be the course, whatever might be the subject I teach. Attending the class itself is for preparation. Then for preparation, another preparation, I don't believe in that policy. Okay. Then 9 a.m. timing for anthropology, new batch. 9 a.m. little bit difficult. I'm considering 11 a.m. That is my dream timing. Because like when I was in Vajiram also, I don't know the reason. 7 years, they have given only 5 p.m. batch. 5 p.m. batch. Because mostly 11 o'clock is prime time, so they use that for GS classes. Optionals are usually 5 p.m. So 5 p.m. means my problem is till 5 p.m. I cannot involve in any other serious work. Why serious work? Say if I have to go for a movie or shopping or some household work. If I go out, there is no guarantee that I can reach the time. And from last 2-3 days, like you might have noticed, I'm being some 5 minutes late. The reason is because I got cured from COVID and sanitizing my home. So every day I get a target of 1 floor, like today ground floor, today first floor, something like that. So I'm unable to reach the target. 3 floors. It is a penthouse. I have recently taken it. So not penthouse, I'm sorry. This is a villa. It's 3 floors. Yes, I was teaching anthropology. I was the only faculty in anthropology. Answer writing that I still did not start, but I have a plan of starting. So I'm starting it in the morning. So I'm trying to wind it up by 2:30. But a little bit is pending in my body. I got so much scared that if one place is contaminated, what if the virus comes from there to here? So such thoughts are coming. So it's okay, let it be 5-10 minutes late, but so that is why I'm being a little bit late these 2-3 days. So if I'm not satisfied with the sanitization process, then what happens? I will again re-sanitize the complete room. So for which I don't have the sufficient patience. So such a frightening and scary situation. Life became so difficult. Yeah, actually when we shifted to this place, 2 people came for fixing the air conditioner. So that day he even sneezed, but when I asked him, did you sneeze? He said, no, I did not sneeze. Probably that could be the reason. Now I'm reading more about reinfection. What is the possibility of reinfection? Are the reinfections milder or more severe? Yeah, he might be not scared with my expression, did you sneeze? Yes, I should take, but they said I should take after 2 months. I should not read, but what to do? Okay, yeah. So, now coming to this, as of now, human genetics means this one. Okay, then twin method in human genetics, this is something that we have to discuss. This is something that we have to discuss that we will see in the next class. Meanwhile, let us complete another small topic. This is something that you know, okay, but here I will add some more points to it. Do you remember chromosomal aberrations? What was the diagnostic method? What was the diagnostic test for chromosomal aberrations? And why we spoke about that sanitization is why I don't like 3 o'clock, 5 o'clock classes. Because if it is 11 o'clock class, okay, I will complete the class and the rest of the day I am free to do something. So I always like morning classes. 9, 10, 11, that is quite comfortable for me. So karyotype analysis was the diagnostic test, right? Yeah, and I was speaking about that. In Vajiram, optional class was always 5 p.m. Okay, then I enjoy shopping and eating. These are the 2 things I enjoy as such. So if I have to go for shopping before the class, no guarantee that I will reach the class by time. And if I have to go after 7:30, in Delhi, I don't know why, they start closing the shops from 8 o'clock itself. 8 p.m. itself, they start closing the shops. In Hyderabad, it is not like that. In some seasons, they keep the shops open throughout the night also, if it is some festival season. But in Delhi, at least, Karol Bagh shopping, I could not go because of that schedule. I could not go means I went many times, but I could not go so many times as I wanted. So now discussing karyotype analysis. But those are all the golden days. Golden days. We used to just freely go out, eat, shop, just a walk. So many things that we have done. Now no freedom at all. Curfew, lockdown, mask, gloves, sanitizer, so many things. Yes, that will surely come back. Then we have to literally enjoy him. After that, you will watch me taking the classes in different places. See, today I'm in a park, today I'm here. So I will not take the classes from home then. Okay. So now, that karyotype analysis is given here in 9.1. In 9.1, that is a topic. Okay. So, you know so many points about this, so we can quickly discuss this one. Okay, and after that, twin studies is a proper topic that we will discuss in tomorrow's class. Karyotype is arrangement of chromosomes based on their size and shape, you know this? And do you remember, let me check your memory also. What are the three sizes? Three sizes? Large, medium, small. Okay, now I will ask the question for your answers. Shapes? Shapes? There are 4 shapes. What are they? What are the names? Metacentric, submetacentric, macrocentric, telocentric. Very good memory, very good memory. Then analyzing the karyotype of individual for the diagnosis of chromosomal aberrations is called as karyotype analysis. And this is the structure in which you can write the answer. Okay, answer structure also. Analyzing the karyotype of an individual for the diagnosis of chromosomal aberrations is called karyotype analysis. Size based on size, small, medium, large. Okay, based on shape, 4, metacentric, submetacentric, macrocentric, telocentric, and you can draw their diagrams also. Next, we have banding techniques. Did we discuss banding techniques there? No, right? Okay, very good. Now let us see. Do you remember chromatin is 2 types? And what are they? What are they? Euchromatin, heterochromatin. Do you remember euchromatin is loose? Heterochromatin is tight. So, let us suppose if you put one common sense question, if you put some color, and you ask me why do we put color on the chromosomes? To see them. Because chromosomes are originally transparent, we cannot see them. So if you color them, then you can see. That is called staining techniques. Staining, isn't it? We also stain the bacteria to see them. Okay? So, when you stain them, if you put any color on them, which part of the chromatin do you think will stain darker? It means if you put a green color on that, which part will stain in dark green? Which part will stain in dark green? Very good, heterochromatin. Okay, this stains dark, this stains light. Reason is, heterochromatin is tightly packed. If it is tightly packed means what? Densely packed, isn't it? Per unit area, you will have more chromatin in that. Per unit area, you will have more chromatin in that. So, when you put the color, more chromatin will take more color, so it stains darker. Is the logic clear? So, if you stain any chromosome, in the chromosome, wherever you have heterochromatin, there it stains dark. There it stains dark. And wherever you have euchromatin, there it stains light. Clear? Clear till now? This is one point that you have to remember. One more point. Do you remember constitutive heterochromatin, facultative heterochromatin? Swati, if this is first class and if you don't have a background, it is very difficult to follow. Very soon I will be starting a new batch also. One alternative is you can either join the new batch. If you don't have a time, you can follow the recorded classes. Whenever you have doubts, join the live, ask your doubt, clarify your doubt, leave. That is a better process. Okay? Join the live occasionally just to clarify your doubts. Okay? New batch June. Date, time not fixed. June. Probably late June and timings in the morning 11 o'clock. Achha. So we can ping it also. I don't know. How? Okay, that I will try to get to know from others. It's okay. Okay. So, facultative heterochromatin means it is sometimes euchromatin, sometimes heterochromatin. Constitutive is always heterochromatin. Isn't it always right? And also, do you remember we have constitutive heterochromatin at centromeres and telomeres? Do you remember? The portions that I have shaded in red, they are always tight. Constitutive heterochromatin. Why? To maintain the structure of the chromosomes. Adish, know for what? To maintain the structure of the chromosome. Because at the centromere should hold. Okay. Now clear? The centromere should hold the 2 chromatids together, right? Centromere is the place where the 2 chromatids are held together. So Ananya, could you recollect it now, at least when I said that? And telomeres, they are like caps, for example, a cap for a pen. Okay? So there it should be tight. Okay, then in between also, for example, do you remember euchromatin is active functional, euchromatin is functional, heterochromatin is non-functional. So in between also, wherever you have junk DNA, there it can stain dark. Did you understand? Wherever you have junk DNA, there it can stain dark. So, when you stain a chromosome, it looks as if it is banded, isn't it? Bands. Light and dark band. Light band, dark band, light band, dark band, isn't it? Say, therefore, it is called banding techniques. Banding techniques. Got it clear? The staining techniques are called banding techniques. Okay? Then, why should we use the banding techniques? What is the use of it? Why do we need the banding techniques? Do you remember we have only 3 sizes and 4 shapes, isn't it? 3 sizes, 4 shapes. Right? So, do you agree that there can be many chromosomes and moreover, most of the chromosomes are submetacentric? Not only that we have only 4 shapes, most of them are submetacentric. So, is there a chance that two different chromosomes both can be large submetacentric? Did you understand my point? Say for example, chromosome number 1, chromosome number 2, both are large and submetacentric. Then, how do I differentiate between chromosome number 1 and 2? How do I know whether it is chromosome number 1 or 2? Did you understand? It means, can we identify the chromosomes only based on their size and shape? All of you please, can we identify the chromosome? Identify the chromosome means when you see a random chromosome, can you say whether it is chromosome number 1 or 2 or 3 or 4 or 5 or X or Y? Which chromosome is it? We cannot say only based on the size and shape. Okay? But diagnosis means saying that only. For example, one chromosome is missing or one chromosome, say for example, do you remember Down syndrome? What is Down syndrome? 21 trisomy. So, in Down syndrome, there are total 47. Okay? I counted the chromosomes, there are total 47. But how do I know which chromosome is extra? Did you understand? Depending on that only, I should say whether it is Klinefelter's or super female or super male or Edwards, Patau's, Downs. Do you remember? In all these cases, you will get 47. So, how should I diagnose which disease it is? When there are 47 questions, it can be Downs or Edwards or Patau's or super female or Klinefelter or super male. These many possibilities are there. So, how should I diagnose? For that, I have to say which chromosome is extra. So, the extra chromosome, I should be able to identify which one it is. Can we identify which chromosome? Is it only based on size and shape? No. So, we need banding techniques. This band pattern is unique to a chromosome. Got it? The band pattern is unique to a chromosome. Like our fingerprint. No two chromosomes have the same banding pattern. So, using banding pattern, we can identify which chromosome is it. So, that is the reason why we do the banding. Okay? So, I explained you the purpose of banding. I will stop this class here today. It's 5 o'clock already. Okay, and karyotype analysis is not completed. That we will continue in tomorrow's class. Maybe 10-15 minutes we will be completing the karyotype analysis. Then we will discuss twin studies. That is tomorrow's topic. Down syndrome, 21 chromosome will have same banding. Yes. So 21 chromosome, 1 copy, 2 copy, 3 copy, 4 copy. How many ever copies you get? 21st chromosome will have the same banding. Means extra 21st chromosome will have the same banding pattern as the first 21st chromosome. Homologous chromosomes have same banding. Homologous chromosomes means what? The banding pattern is unique. Banding pattern is unique for a chromosome. Means for 1, 2, 3, 4, 5 is different. Okay, banding pattern of homologous chromosomes is similar. Because one comes from the mother, one comes from the father. In whoever you have it, that chromosome will have that banding pattern, isn't it? Okay? Thank you so much, Akhil. Fine. So, that's all for today's class. Thank you all so much. Bye-bye.